So our first speaker, Johannes Wirz, a doctor from Goetheanum, I think I'm pronouncing that, in the natural science section. After his PhD thesis in molecular genetics on the Hox genes of Drosophila at the University of Basel in 1987, Dr. Wirz joined the Research Institute at the Goetheanum, Dornach. For the past 2 years, he has served as co-leader of the science section until his recent retirement. Also, from 2014 to 2025, he was a member of the board at Mellifera, EV in Rosenfeld, GE, Georgia, I think, and coordinated bee research projects. He publishes regularly, gives seminars and lectures on modern and Goetheanum science, as well as Anthroposophy and the biology and life of honeybees in international settings ranging from the US to Europe and China. So welcome, Dr. Wirz.
Hello, everybody. I feel honored and it's a pleasure to share with you some thoughts about the evolution of science, evolution and genetics in the last 250 years or so. In the preparation, I realized that what I want to tell you is somehow also part of my scientific biography. As you have heard, I started as a molecular geneticist at the University of Basel and later on learned to become a Goethean scientist. Now, I hope that I do not overload too much my contribution. So, in a nutshell, I will tell you what I would like to share. We will start in the middle of the 19th century with Lamarck and Goethe, Darwin, and we'll see that in those times, these concept of purpose, of higher development in evolution, directional evolution, um, agency, and phenotype have been very important concepts. And I would like to show how these concepts were destroyed in the course of the development of genetic and evolution in the 20th century. And that, in my eyes, 1950 is a marking point and birthday of molecular genetics. I would like to show you the progress in this field, and I will end with a postulate that says that modern molecular genetics is at the threshold to rediscover all the concepts like purpose, agency, etc. they have lost on their path towards the future. And I would like to end with a short outlook about the necessity to introduce these concepts of purpose, agency, higher evolution, and phenotype for the sake of the future of man and Earth. Now I would like to share my screen with my presentation.
Sure, Dr. Wirz, you may just have to send a request and then I can approve it from my side. You should be good to go now.
Okay. Yes, here it is. Is it possible to make it full screen? I think I can do it myself. Yes. Please don't hesitate to ask questions or interfere if something is not really is difficult to understand. In contrast to the title, I have written down in the program before, I have added on the loss and rediscovery of plant and animal and human wholes. Yes? So I would like to start with Lamarck. And I dare to do so because in 1882, the eminent evolutionary scientist Ernst Haeckel has written an obituary for Charles Darwin, and the title of this obituary has been Lamarck, Goethe, Darwin. So Haeckel saw these three thinkers somehow in connection or in a row with respect to evolutionary thinking. Now, Lamarck's theory, he has been the first thinker, modern thinker about evolution, and his theory had three different building blocks. The first, spontaneous creation. This is shown here on the figure by these dots that start some earlier or later in time. The second was, and this is shown by this straight line, that in every organism that comes into being, there is a capacity and aspiration for higher development. And the third aspect of his theory has been this can only be achieved by the inheritance of acquired characters. Of course, we know Lamarck has been out of science for most of the time until today. The second thinker I would like to add, and in contrast to Lamarck, who had purpose and directional evolution, Goethe was not, well, he was thinking about evolution, but his main point is that he put a system of thinking, presented a system of thinking that took into account that nothing can come into being, no plant, no animal, if there are not two aspects helping to bring it into life. And he called this the so-called double law. And the meaning of double means it is not two. It's like two sides of one coin. So if we see an organism, we must take into account its inner nature. Goethe called it also archetype or agency, and it can only come into being in an outer world. And he said there is a law of outer circumstances, outer conditions that help to bring organisms, plant, animals, maybe man, into being. And then he said an important thing. This inner law of inner nature or archetype constitutes the organism. The law of outer circumstances is modifying the shape, the form, the gestalt of the organism. And then he outlined that in order to understand the law of these outer circumstances, he needed a couple of so-called, he called them auxiliary sciences, from history of nature to morphology in a strict sense. And I would add, today we should have genetics, evolution, and maybe behavioral science as well on this side of the outer circumstances. And you can well see that if you cut off all the upper part of the organism, Goethe somehow anticipated the development and evolution of modern science. So, Lamarck purpose, directional evolution, and inheritance of acquired trait. Goethe, there is an agency in living nature that brings living beings, plant, animals, and eventually man into being. And then came Darwin. And we all know that Darwin has developed an evolutionary theory where random variation is a very important thing. And at the same time, selection is another important thing. And then he said, and now I would like to read it, but I have all these people sitting there. And then he said in his book on the origin of species, the following words: to suppose that the eye, with all its inimitable contrivances for adjusting the focus to different distances, for admitting different amounts of light, and for the correction of spherical and chromatic aberration, could have been formed by natural selection seems, I freely confess, absurd in the highest degree. What do I want to say with these words? Although Darwin was deeply convinced about his theory, he was also intelligent or wise enough to say that the theory was not a theory of everything. So he still had something in mind that says there are certain traits and properties that we can never ever explain by my evolutionary theory. I would like to, the next thing, now we are already in the 20th century, I would like to mention is Ernst Mayr. Ernst Mayr was one of the founders of the so-called synthetic theory. And one aspect of this new evolutionary theory was the reconciliation of evolutionary thinking and genetic thinking. And then he said in his book, The Growth of Biological Thought, it was perhaps the greatest contribution of the young science of genetics to show that soft inheritance does not exist. And soft inheritance means the inheritance of acquired traits. It does not exist. And we see an amazing reduction from Darwin to Ernst Mayr. At the same time, almost the same years, there was another very influential thinker, Conrad Waddington in Edinburgh. He was an embryologist and evolutionary biologist. And he said, I don't want to doubt about Darwin's concept of random variation and selection. But there are certain traits we can see in organisms that never ever could have come into being by just random mutations of whatever, and then accumulation of random mutations. And he took the example, and you can read this if you want in the book of Soltax. You can download it on the internet. He took as an example the forearms of a gibbon on the left side and the forelegs of a pangolin on the right side. And you can imagine first, how should it ever be possible from a maybe common ancestor to go in random steps towards gibbon-like limbs or pangolin-like limbs? One thing. The second thing he was also clear is that not only the limbs are adapted to a special life and behavior, but the whole anatomy, physiology is so. So for him, it was impossible to think that these very important traits could have come into being by random variation, random mutation, and selection. And I wanted to show you that this, because in this mainstream thinking, where everything was purposeless, no agency, molecules and molecular processes, then there were also thinkers, and I will show further thinkers in a minute, that had a precise and a very logic arguments against. Now I would like to come to another actual example. It has been published in 2014 by Emily Stanton and collaborators, and they made a very easy experiment. They took the baby fish of a lungfish, Polypteros, and one group of fishes were raised on a solid ground, and the other groups, sisters and brothers, were raised in water in an aquarium. And then, after the development, they looked at the behavior of the fishes grown up on solid ground and the fishes grown up in water. And they could easily see, and it is very nice to look at the videos they made, that the baby fish grown on solid ground were quite nicely adapted to the solid ground. They could move faster, they could move with less energy, etc., compared to the siblings that had been grown up in water. And then they looked at the anatomy of these lungfish, and they could see that in this short period of living on solid ground had tremendous changes on the whole vertebrates. They could move the head higher on. They had a collarbone that was very different from all the others. Just briefly, one generation living in a different environment leads to substantial and integral anatomical and physiological differences. And then the question, of course, is it possible and how is it possible to fix such differences? I will come to that later. And now I will come to the birthday of molecular genetics. And for me, the double helix of DNA, this means the structure of our genetic material, seems to be the entry point. And I show here a copy of the original presentations of Watson and Crick of the double helix of DNA in Nature, in the prestigious journal Nature, 1953. And first, I want to say, from an aesthetic point of view, it's a very beautiful structure. This seems to be so clear. Second, in order that this structure comes into being and is stable, we have to know that there are only 4 building blocks of this genetic material, the so-called nucleotides, adenine, thymine, guanine, and cytosine. And this structure only is possible if a situation where on one strand, let's say the left strand, at this position where my arrow is, there is a G, then on the other position of the second strand, there must be a T. And if here is a G building block, guanine, then here on this strand, there must be a C building block. You understand? So there is a pairwise restriction to the organization of these 4 letters of the genetic alphabet in the DNA double helix. And the consequences of this restriction is very important. If you would open this double-stranded helix and every organism, every cell must do it always before cell division, because before a cell can divide, it must make sure that the genetic material is replicated. If not, it would be lost in one cell and only be in the other. And you can imagine the strand goes up, and now one strand is kind of the blueprint for a new strand. And when this is added, when this is an A, the new strand must have a T. When this is a C, the new building block added on the second strand must be a G. This way of replication is called semi-conservative replication. And this had led to the idea that this genetic material doesn't need anything except a certain chemical character in order to build and rebuild itself. And out of this, this notion, there was the idea, they called it, it is not later on called by somebody, it was Watson Crick that coined the term the central dogma of genetics. And what I want to show with the circle is the DNA is self-sufficient in order to make new replicas before every cell division. And the second thing, I cannot go into details, says there is a flow of information going from DNA in the nucleus of a cell to RNA, kind of a messenger, where part of the DNA is transcribed, and out of the RNA, in a process called translation, proteins are built up. And this central dogma of genetics has become kind of ideology. Next slide. The first thing we see that it is not just a chemical process adding building blocks or these letters A, T, C to a strand that gives a blueprint. Look at the figure on the right side. You see here genome size. This means how much genetic material is there. We can say here are primitive organisms. Here at the end here is man. And you can see the smaller a genome is, the less the genetic material is, the higher is the mutation rate. The larger it gets, the lower is the mutation rate. And it's very easy to understand why this must be so. And at the same time, it is an indication of kind of an organismic wisdom inbuilt in more simple organisms and more complex organisms. Imagine a bacteria would have a slow mutation rate, then this bacteria could never ever evolve. And imagine that man with a large genome here had such a fast mutation rate as a bacteria, it would not evolve either, it would just die out. Because more than 90% of all mutations that naturally occur are eventually deadly mutations. So what I want to show you with this picture is there is much more than just a chemical process that helps to replicate DNA before cells are dividing. And the other thing that also came out of this early molecular genetics was that there are genes. This is a sequence of information that somehow tells the cell how to make protein. And for a very long period, very long is maybe not right, for decades it was believed that one gene makes one protein and the organism is no longer whole, but is just the addition of different building blocks. And to show the problem of this view, I have allowed myself to add a picture out of a study we did in our institute. It is clear that with the advent of molecular genetics, there was also a technological side. And since the mid of the 70s, geneticists have learned to genetically modify organisms and to develop what is called today biotechnology. And on the basis of Goethe, you may remember that I said, if you change something in the outer conditions, the organism as a whole must adapt to this change. We were convinced that this is also true with respect to genetic manipulations of organisms. And I don't want to go into detail, I just want to tell what you see here is a pintia potato growing as it does in nature. And on the left side and on the right side, you see from the same mother plant, potato plants that have been genetically modified. I just take this one here for the sake of time. In this plant, by genetic engineering techniques, a gene from the mistletoe, viscotoxin, was introduced, and it is known that viscotoxin can fight late blight, a tremendous plant disease in potatoes. I must say that the target to have this blight-resistant plant did not work, but we saw many non-target effects. And you can see here, non-target effect is slow growth dynamics in this plant. The plant remains small and has over the whole growth period, and the morphology of the leaves is very different. And now not to see, this plant never ever make flowers. And now you must imagine, consider the genome of potato to be a book with written letters A, T, T, C, G, G, C. This would be a book with 200,000 pages. If we say we have 3,000 signs on every page. What the genetic engineer is doing, he's adding 3 pages in a book with 200,000 pages. And you can imagine it would take us years and years and years to find where these 3 wrong or new pages would be. So somehow it is intuitive to say, well, there can be only one effect. And at the same time, we can learn that even the smallest change in outer environment results in an integral reaction, not only resistance, but also in growth dynamics, in leaf morphology, in flower morphology, etc. I hope you get this point. This one gene, one protein theory is correct, but the effect of one protein and one added gene is always integral and manifold. Now, next, I would like to show you a few persons that have helped to reshape the concept of molecular genetics in the last decades. And it is not my favor for beautiful women that has led to these persons, but it has to do that for reasons that we also could try to understand. Ladies, women were more open, more sensitive for new ideas. And the first one I would like to say a few words is Barbara McClintock. In the 40s of the last century, she discovered jumping genes in maize. Nobody could understand, and it was only in the late 60s when Charcot and Monod in France discovered the same genetic elements in bacteria, they called it transposons, and they realized that organisms have the capacity to enhance mutation rate if it is necessary. So you see, there is also, again, an organismic wisdom to tell an organism or a population, whoa, things are getting difficult, we must look for ways to secure our survival. The next 2 ladies, Marion Lamb and Eva Jablonka, have a lifelong worked on epigenetics, and they have published a very nice book where they outline how they believe evolution takes place and comes into being. And the book has the title Evolution in 4 Dimensions. The lowest dimension is Darwinian evolution. They do not doubt that it does exist. The second dimension is epigenetic. The third dimension is behavior. So animals that move around can change actively the future anatomy and physiology. And the fourth dimension related to man is called the symbolic dimension. And we know in our culture, we have every tradition is, so to speak, what we have acquired, we give to the further generations, and we can say we do not have to invent the wheel every time anew because our ancestors have done. And then the next lady, Mary Jane West Eberhard, she has been an insect physiologist, and she wanted to know how it was possible that insects with the same genetic material could become differently in their development. And the most known example is, of course, the honeybee. We know worker bees and queens share the same genetic material, but through a special diet, out of an egg and a larva, a queen can hatch. And if you change the diet, then there will be a worker bee. And then in her study, she realized that such events are much more frequent than just in the honeybee. And she has written also a very important and seminal book with the title Developmental Plasticity and Evolution. And there she comes to the conclusion, and I quote her, in evolution, the genotype is following the phenotype. Now, genotype means the totality of genetic material. Phenotype means what we see out there as plant, as an animal. And you can easily see that in the classical molecular genetics, the genotype was governing the phenotype, and she says the opposite. And then I have here added Susan Lindquist. She has been in Cambridge, Massachusetts, and she was among the first to understand what the so-called heat shock protein would do. And they were called molecular chaperones in the sense that they could hold under stress situations vitality important proteins in their shape without getting destroyed by heat, for instance. And the last lady I would like to mention is Isabel Mansui. She is the head of a lab in neuroepigenetics in Zurich, in Switzerland. And she has made a very important contribution towards the ways how traits that are developed in a childhood can be genetically fixed. And she has shown, I say it in one sentence, that if you stress male mouse pups by taking the mother away for 2 to 3 hours for a couple of days, these males will give rise to offsprings in the next generation that without this stress still show the stress of her father. And she could show that this could be transmitted over 4 generations. And then it was being very aggressive opposition against these findings. He could show mechanistically that this really was the case, and I can't go into details. But I would like to hook on this term mechanistic. All these organismic wisdom in epigenetics, in whatever we have learned from these ladies, have been looked with a very critical view because it was said they don't know anything about mechanisms. But, of course, we must say there are facts, and behind every fact there is mechanism. And at the moment where you talk about mechanisms, you forget that it has to do with an inborn inherent wisdom, and you just say, well, this is because they are the molecule and the molecular processes. You understand, by this narrow view, you lose animals and plants as wholes, and wisdom and purpose as wholes out of view. Now, let me go to the next slide. I think it is because of this figure that I have been invented to this conference. And here I would quickly try to say what is behind all these different arrows. You remember the first thing was central dogma. There's a DNA self-sufficient and a one-way street from RNA to protein. And because it is one way, the only way to get any changes in evolution, let's say mutations, you need random events. And then, as I have said with Barbara McClintock, these mutations or transposons have been discovered that could explain that the organisms themselves, plant and animals, bacteria, regulate mutation rate. The second is we have seen, and this has been the work of David Baltimore, he has recently died, who could show how RNA is reverse transcribed into DNA. And the work of Isabel Manzui has shown that regulatory RNAs can find their way from the brain, let's say, to the germ cell of an animal, we have seen that one sequence transcribed from DNA can result in up to 40 different RNAs. So this is called differential splicing. And we have seen also with Mansouri and others that there are now known enzymes, proteins that can add substances to the DNA without changing the core building blocks. And the one substance is methylases make the chromosomes tighter, so these genes in this region cannot be transcribed. So inactivation and acetylases make the chromosome looser. So these sequences are open for gene transcription. And the last thing we have heard, these chaperones worked on by Susan Lindquist. What I want to show with this picture is DNA is important, no way. It's a very important molecule, but it can only be important in the context of very many different dynamic situations. And there must always be a flux from DNA out into the cell, in the cytoplasm, but there is also always information and material flowing back to the cell of the nuclei. And please keep this picture, a cell as a whole cannot depend on one single molecule for its existence and development, but it is always kind of an orchestra of very many different instruments. And now I would like to come to the Human Genome Project. This has been a turning point. 2003, it was presented and it was heralded as the book of life, this human genome, or the Holy Grail of biology. And when it was deciphered, 3 billion letters, A, T, C, G, T, 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 C, A, an enormous encyclopedia, there was kind of a disillusion. Because first, nobody could read the book. Second, it turned out that mouse, Drosophila, and man had the same protein coding genes. So where is the difference between mouse, man, and Drosophila? So nobody could understand. And then it was realized that there are some 98% of so-called junk DNA. And it was asked, why on earth an organism would keep so much junk in the chromosome that is not needed? Of course, it is needed. And later on, it has been shown that in these so-called building block sequences, there are thousands and maybe millions of small RNA molecules to regulate, to orchestrate the whole dynamic process of a living cell. And in addition, it could be seen that 50% of our DNA has been somehow integrated in the course of evolution from bacteria and viruses. And the picture that appears is that nature, man, and all the organisms around is one big whole or one whole. Yeah. And now I would like to throw a view on what is to come. And beside this genomic and transcriptomic, etc., science, there's a new discipline coming up called microbiome or microscience. And with technical possibilities I cannot describe, we are now able to look at all the microbes in our body from birth to death, in different dietary situation, in different, for instance, milk feeding or non-milk feeding, in different geographies, etc., etc. And we can realize, and I didn't believe it, there are thousands of different bacterial types, not only on the skin, in our mouth, and in our metabolic system, but every single organ has different bacterial strains, types associated, and all together in a high diversity and variability, these are the bugs that help us to be resilient and healthy. And
Dr.
the very
Wirz,
same is true when we look in the non-human world. We
Dr. Wirz,
find,
I'm sorry.
yes,
Just as far as the clock goes,
I still have 5 minutes.
we could give you another 10 minutes for the speak. I just want to let you know another 10 minutes. Is that okay?
Oh, okay, thank you.
Sure.
Thank you. And so the very same situation is true when we look outside into nature, be it air, be it water, be it the soil. And again, there are thousands and thousands of different bacterial species there, or types, we should say. It's difficult to talk about species. And in combination, in connection, they are able to help plants grow better, to detoxify the soil, for instance, to bind toxic heavy metal, for instance, as shown here, etc., etc. And interestingly enough, some of these bacteria are taken up by the plants. We take the plants, we feed on these plants, we eat these plants, and the bacteria, important health and resilient carrying bacteria, enter our body. And so we have an amazing expanded view on all levels, the cellular level, how our inheritance or genetic material is organized. On the organismic level, it goes only in a connectedness of different organs, different cells, etc. And also on the large level, the health and resilience of man on Earth is interdependent on each other. I think this is a very important message. And so for the last minutes, I would like to go a little bit further with this message, because we are still, and now I try to come back to, I will not show any slides, but I would like to show you that, or to share my opinion that it can expand this connectedness. And I want to say, first, we can discover that there is kind of a purpose in all living beings. Second, we see that there is an amazing interconnectedness. So diversity, community, is it? We are not single warriors on a battlefield, but we are working all together. And we also can ask, who is orchestrating this amazing symphony we see inside our body, in animals and plants, and around the whole earth? And I think this is the moment where the concept of agency must come back. We cannot find agency. Goethe already said, I see ideas, but not with my eyes. They are invisible, but I see it with our thinking. But we must take into account that such an agency is there not only in a human head, a human consciousness, but ontologically out there. And I would like to end with an idea I have found with the philosopher Hans Jonas, who, interestingly enough, has written a book as a philosopher, The Phenomenon of Life. So I would say we are coming back through modern science to rediscover purpose, sense, agency, and the phenomena again. And Hans Jonas, in this book, it is a book with a couple of hundred pages, he says, don't want to doubt about Darwin evolutionary theory. But I suggest that there are two ways of looking at evolution. Darwin did it from the past to the present. I would like to do it from the present or the future to the past. You see, vice versa, or opposite direction. And then he starts to ask, what is the most important quality, property, trait connected to us human beings? And he says, it's freedom. And then he goes on and he says, now we must learn to discover pre-forms of freedom in plants and animals. And then he comes to the conclusion that the first sign of kind of a freedom is metabolism. In contrast to inorganic nature, where just things that come into being also decay, we always in our metabolic system destroy food, outer nature we could say, in order to maintain our form and gestalt. A next step is then with animals, they have the same ability like plants, but in addition, they can see, perception, they somehow can feel emotion, and they can move, motion. So, vision, emotion, motion. And then in the next step, man has everything plants also show, has everything animals also have, and in addition, he is able not to follow his instincts, but to look into the world and to take the world as a picture. So he says, Homo pictor. And we all know, be it from our life or from philosophy, that pictures do not oblige to do something, but they leave a space where freedom can come into action, and then it helps. We decide, hopefully free, I do this or this. A dog who sees a piece, a bone, cannot think, should I take it or not? He just jumps and takes it. It's maybe a bit anecdotal, what I try to say. But you understand, you can look to evolution from two sides. And the aim then, and Hans Jonas is very sure about that, if I look it from this point of freedom, then I must say there must have been an aim or teleology in evolution. And I would like to cite one sentence out of this book, and you will understand why I have said that. In the epilogue, he says, after what he has written on some 250 pages, a philosophy of mind comprises ethics. This is what we believe is true. Ethics is connected to our intellect, to our self-consciousness. But our philosophy of mind comprises ethics, and through the continuity of mind with organism, that's what he has shown, and of organism with nature, ethics becomes part of the philosophy of nature. I read it again. Yeah, for me, it is very, very important. A philosophy of mind comprises ethics, and through the continuity of mind with organism and of organism with nature, ethics becomes part of the philosophy of nature. So, all of a sudden, ethics is no longer just a question between us human beings, but it is part of a potential capacity that can be found back in nature. And if we want to act and behave in a moral, ethical way, we must act and behave according to what we detect in nature, sharing, working together, etc., etc. And then one important thing is, and I would like then to end with that, before we can do that, we have to work on ourselves. And there is a simple word of Mahatma Gandhi who says how we can come to this point. And he once said, you must be the change you want to see in the world. So We must do something in order to see the ethics out in the nature. We must learn to live interconnected in companionship, in respectful or even loving way, in order to see and find and bring respect, love, and connectedness into the world. Thank you very much for your attention.
Thank you very much, Dr. Wirz. You offered an excellent history of science, starting with Goethe's double law of the outer circumstances and inner nature of the organism, touching on Charles Darwin's open-mindedness about the limits of natural selection, moving on to acknowledging some scientists who had some pushback about Darwin's ideas, like Waddington, acknowledging the beauty of the double helix DNA structure and its semi-conservative replication, leading to the central dogma of molecular biology of a linear causal sequence of DNA to RNA and protein, which results in one gene, one protein, is a correct analysis, but that the effect of editing one gene is manifold. Then you moved on to presenting to us the innovations of several women in science who were showing that beyond what Darwin laid out with his theory of natural selection, there are other causes that influence evolution. And so one of those ladies were Mary Jane West Eberhard, claiming that the genotype follows the phenotype rather than the opposite understanding, which was traditional. You talked about the need to balance mechanistic explanations within an organismic context, the need for contextualizing genetics within biological activity of higher levels of organization of cells, tissues, and the organism. And you concluded that portion with the Human Genome Project at the turn of the century, which was very wonderful with the achievement of documenting, of sequencing the whole human genome, but also showed the lack of fulfillment of some of the promises that resulted from that sequencing. You concluded with an ecological vision of science concerned with the interconnection and wholeness of a scientific endeavor studying nature and seeking to further understand the agency in nature's splendor. It was very instructive what you said about viewing evolution rather from the past to the present. Better we should start from the present and move our way backward. And also, lastly, perhaps we can talk about this more in the interdisciplinary dialogue at the end of the conference about this idea of gestalt, or the nuances behind biological form, and why that's significant for understanding various biological processes, as well as agency and the relationship between organisms. So we thank you very much. We have about 10 minutes for Q&A if anyone has anything to ask for Dr. Wirz.
Thank you for this very nice summary. Thank you.
I'm trying to figure out the hand raising function on this, but maybe I'll just go ahead and speak.
Organic works.
Okay. Thank you, Dr. Wirtz. I found this a really fascinating talk, and appreciate your work very much. Gosh, so I mean, you talked about agency in interesting ways. I'm going to talk later about Hegel and some of the influences of 19th century biologists on his view of systems, which he goes on to apply to lots of areas besides organisms, besides natural organisms, as he characterizes the state as an organism and things like that. And then I'm talking a little bit about, well, some scientists who criticize this Hegelian view, and Tansley in particular, who proposes the ecosystems concept in a paper in 1935. And I guess in that context, I want to ask about agency, as it relates to something like ecosystems. You were talking about agency at the level of cells and agency, the pre-molecular level, and then of organisms. Um, and the fact that they're influenced by this environment and the very genome could be affected by and perhaps changed and perhaps have elements fixed because of environmental conditions. I'm wondering if you just know more than myself. I'm a philosopher, not a biologist, of work that kind of looks at, I don't know, these ecosystemic wholes from an eugenic perspective. I mean, we can see how they interact with each other. We tend to think of agency, I guess, at the organistic level or cellular level. And I know it's an unfair question. It's not what you're looking at, but I wonder if you happen to, uh, given your rich body of research, know of anything that addresses that type of question.
Um, I would like to make three remarks. First, Hegel and Goethe have been friends,
Right, right.
and Goethe had the impression that the way he was thinking about organisms was the same way Hegel was thinking about thinking. And the agency, I try to say, the idea of the archetype by Goethe is not it's maybe a weak translation to say agency. This is something that has to do with our mind. And if we go back to Goethe, it has also to do with the mind or the idea in nature. So this is this invisible property, and maybe if we would say it in two words, it's the question of mind and matter. And in between, everything develops and evolves. And now coming to your question, the interesting thing at the moment is I don't know whether there's something behind all these communities, be it bacteria, be it animals or plants. But one thing is for sure, we can no longer identify the right bacteria or the right plant in an ecosystem. And it is as if they would work together to create resilience, to heal the ground, to heal our body. So it's a very new concept that it's not a single one, like one gene, one protein, one bug, one toxin, or like that, but it is the bacterial or microbe community as a whole, be it in water, be it in the soil, be it in animal, plants, and humans, that make the resilience and make ecosystems stable, secure, and somehow able to develop into the future. I don't know whether I have answered your question.
Yeah, thank you. That addresses it for sure. Thank you.
Okay, so we do have another 2 minutes in the Q&A session. If anyone has a question, please feel free to turn your camera on if you like and ask. No need to raise the hand with the application. I have a question myself, but I'll hold unless nobody else has a question. So Dr. Wirz, my question is, do you think it's fair to use the word dialectic to characterize the feedback between the DNA, RNA, and protein that you showed?
Mhm. I have written a personal mail. As a Goethean scientist, I'm, how should I say, accustomed to talk about polarity. But reading some of the texts you have sent, I realize that you are talking about thesis, antithesis, and synthesis. And for me, see the same structure back in Goethe's way of looking at nature. On one side, idea, mind, or spirit, on the other side, the matter, and together they create these endless forms, beautiful forms, resilient forms, etc. So I would say maybe it is kind of a synonymous to dialectics. And as I said before, I believe that the friendship between Hegel and Goethe had to do with the fact that they felt, maybe not explicitly, but implicitly, working in the same direction.
Very nice. Um, so thank you again for
Thank
your
you.
talk and your insights. We hope that you will join us for the interdisciplinary dialogue at the end.
For sure.
Uh,
Thank you.